Hello, everyone, and welcome to phyloseminar.org. The current theme is evolutionary ecology, and this is the first talk in a series in 3 talks in that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Chris Wheat, an associate professor at Stockholm University. Chris did his PhD at Stanford. Then, the road from California to Stockholm took him through 2 postdocs, the first in Germany at the Max Planck Institute for Chemical Ecology, and the second split between the University of Helsinki and Penn State University. Chris's work uses an ecological and evolutionary functional genomics approach to study plant-insect interactions, life history trade-offs, and seasonal plasticity. Welcome, Chris, and thank you for participating.
Excellent. Well, thank you for that very nice introduction, Eric. And it's really wonderful to be here. In fact, I thought I'd start off with a little slide and just say, really, thanks for the invite. And it's nice to be part of this long-running online seminar series and to have more connection with the Society for Systematic Biology. It's one of my favorite journals. A little bit about me before I get started. I just want to say that we do a lot of things in my lab. You can call it the wheat lab, but we do not study wheat. We use butterflies, actually, and a lot of different insect models to study the chemical war between insects and plants, specifically gene family evolution and butterflies in response to glucosinolate detoxification of their plants. We're also really getting deep in the transcriptome, looking at alternative splicing in plastic phenotypes, and how we can use that to try and get insights into how genetic accommodation is evolving. And one of the last, I guess, lines of research, major research in my group, is looking at the causes and consequences of alternative life history strategies. And that's the focus of my work today. That's work done primarily by one of my PhD students, Kali Tunstrom, and really a big shout out goes to him doing a lot of the heavy lifting here, along with a lot of other collaborators, which I'll mention later. My view of phylogenetics, and I'm trying to tailor this to the audience that might be here at the Philo Seminar series. I have a very kind of integrated view, I think, of how I use phylogenetics. I primarily use genomes, and with genomes, I can reconstruct phylogenies. I can look at molecular evolution. I can get at the basis of adaptation, and I can reconstruct population dynamics. So when I think about phylogenetics, I think about phylogenomics. And then once you have genomes, you can actually do a lot. They're incredibly powerful. And between these kind of 4 simplified blocks of disciplines, if you will, between phylogenetics and population dynamics, we can think about the multi-species coalescent, incomplete lineage sorting, introgression, speciation. Between phylogenetics and molecular evolution, we have lots of variation in the rates at which things evolve across the genome, and larger scale events like gene birth death dynamics, which I think are incredibly important and challenging to work with. And then between adaptation and molecular evolution and, say, population dynamics, we've got lots of tests of selection, evidence for convergence, and the basis of traits that we can get at. And so this kind of view is really where I think the field kind of is maybe at the cutting edge. And it's interesting that for a journal like Systematic Biology that really focuses a lot on phylogenetics and integrating a lot of the new genomic tools to try and answer questions that they've always answered, I think a lot of this stuff can kind of come in and maybe eventually be part of that integrated framework for the journal. And I'm curious to see how far it starts moving in that direction in the future. Today, I'm going to be talking about butterfly wing color evolution. And this is just a drawer that you might see if you pull out something when you go to a natural history museum. And the colors and shapes are astounding. Our understanding of the evolution and development of these traits has made incredible advances in the last decade. And that's kind of where my talk today is going to be focusing. But usually when we talk about butterfly wing colors and their evo-devo, we're thinking about heliconius butterflies, which over the past decade, there's been an incredible amount of understanding developed in this system. These butterflies are primarily known for their mimicry rings down in the tropics where different species end up converging on their color and pattern. Some examples are shown here. And on the right side, I put up this figure from a paper back in 2017 from Von Bellingham and colleagues, where they're integrating an incredible number of studies to kind of demonstrate how across the top we have 4 different regions in the genome of these butterflies that together can be used to create the patterns that fall along the y-axis in all these different rows. And so there really is kind of an evo-devo wing color pattern toolkit that is emerging from this diversity of studies, and it's really quite amazing. But today, my focus is not on camouflage or these type of mimicry patterning studies. I'm actually focusing on color as a costly phenotype. And costly from a physiological sense. I'm thinking about the evolution of color as a trade-off with other aspects of life history. What I'm showing here is a phylogeny of European butterflies. This is a study that came out of my lab a couple years ago. And for each of these European butterflies, about 300 of them, we digitized images of them and looked at their color evolution across a phylogeny. We did this for males and females to look at their rates of evolutionary color change to start trying to provide a framework for looking at how color actually evolves. But when we think about color, or when I think most biologists think about color and how it's evolving and what causes it, they primarily think about melanins and carotenoids that are usually driving a lot of the color variation that we see around us in the world, from the vertebrates and the invertebrates that we encounter. The metallic patterns that we see in a lot of butterflies, such as this morpho butterfly with this metallic blue, that surface diffraction, and it's quite an amazing way to generate color. But today, I'm focusing on this butterfly family, the Pieridae, also known as the yellow, whites, and orange butterflies. And they don't use melanins and carotenoids. They use a Pterin-based color, Pteridines. And here are some examples of different animals that also use Pteridines for their color variation. And these colors range from a yellow that you see in Sepia-Pterin to kind of a red that you see in Drossopteran to Erythropteran for oranges, whites, Leucopteran. So this is the kind of color palette that Pterins can paint, and that's what we're going to be focusing on today. Now, in this butterfly, it's a very good example of color variation from orange to white. And here, this white variation is called alba, kind of like an albino phenotype in terms of it being white compared to the other colored version, in this case being orange. This is a Colias crocea, butterfly found across Europe in alfalfa fields. And in Colias, this alba phenotype, segregating in populations, it's only found in females, and it's actually an alternative life history strategy. And it's not mimicry, and it's not camouflage. Why? Well, a number of studies have shown that the development time is actually faster in these alba females compared to orange. These alba females also emerge from their pupal stage with more fat body. As adults, they have a higher fecundity. Males don't like them as much, so they're not as attractive. And so all of this stuff kind of comes together as alba being a rather complex suite of traits that is an alternative life history strategy that segregates as a Mendelian trait within populations. This is just an example of a very old study from North America looking at Colias, where here you see eggs laid in days after mating, and you see the mean and the distribution and the sample size above each day range. And here you see that for alba, right? And you can see that here in this middle range, as they're starting to reach kind of their more peak egg production, alba is really having a higher fecundity compared to orange females. And this is just an example of how they have a higher fecundity. It's complex, but it's there. So today we're going to be talking about Pieridae, and we're having a slightly different look at color evolution than many people might be thinking about before this talk started. My goal is that I want to build an ecophysiological understanding of how color is evolving in this family of butterflies. And I'm going to be using a phylogenetic foundation to get at how does this color evolve. And my goal for this talk is to really identify orthologous color traits across species. Because once we can establish that, we can then look at all the ecological and physiological differences between these species and ask how that's playing out and interacting with this alternative life history strategy. When we think about different species within the family Pieridae, here's 3 different rows of species. And on the far left, you have alba females and white, of course. You have the non-alba female, which are colored orange and yellow, and then males, which are usually identical to the females. So 3 different genera all have this white morph called alba. And in this summary of a lot of data, Lamary and Morehouse tried to reconstruct the distribution of alba phenotypes across species in pirate butterflies. And what's interesting is that they found a lot of species have ancestral state reconstructions where alba is coded as being polymorphic. Okay, that's all these nodes that have large fractions of green in them. Colias is kind of here in the middle, and you can see there's a blow up of Colias, and there's a lot of species that have a polymorphism for alba. A fair fraction are fixed for the white morph, and some are fixed for a colored morph. Well, one of the questions is alba the same genetic basis across all of these species? If this was true, this clade itself goes back about 50 million years. That would be very old for a shared trait in a polymorphic state across species, right? That's a pretty old transspecific polymorphism. So that's probably not likely. But to some extent, the trait might be doing that. It might have a shared basis, or it might be evolving multiple times, right? So this gets at a fundamental question that I think the people on this seminar might appreciate is, how accurate is a phylogenetic reconstruction in terms of, is this the same phenotype that we're inferring back at ancestral nodes, right? How can we test that? That's what I'm going to try and get at today. Where I'm going to start is looking at 2 species that really are very divergent within the genus Colias. One occurs in North America, one occurs in Europe. You can see that they look very similar in their phenotypes. Both have an alba phenotype. It's white compared to the colored phenotype. They're both kind of an orangish yellow. And so I'm trying to ask now, how did this alternative life history strategy evolve? My goals are to identify the actual locus, the basis of the alba trait itself. I'm then going to place that into a larger evolutionary context, the phylogeny of Colias butterflies. And then I'm going to try and ask if the alba locus is shared across all these species, or whether different species are evolving alba independently. Okay, so this is who I'm going to focus on. Again, the genus Colias has about 80 species, and the alba phenotype is only found in females. It's found in about 1/3 of the species, maybe more. It's a Mendelian trait, so we know that it's a locus that has one large effect. It's autosomally inherited, so it's not on the sex chromosome. So where does Colias butterfly wing color come from? And if you go and open a drawer at the museum, this is what you would see. And if we go in closer, and a little bit closer, closer still, and then we go into one of these actual single cells, these butterfly wing scales are single cells, and we're going to go in, you can see the ridges in there, and we're going to zoom in to right between some of the ridges, and we're going to lose color because we're going into an electron microscope. This is what we see. Now, in these ridges, you kind of see these little grids, these little windows in there. And in there, you see like these little rice granules hanging down. These are pigment granules in the scales with Pteridine pigments in them. Pteridine pigments, again, come in kind of the orange, yellow, red flavors. And the white butterflies that are common in your gardens, going around eating your cabbages, those little granules or pigment are full of leucopterin. They're white, right? So they give this white color. It's a very rich, kind of creamy white color. In Colias butterflies, we see something quite different. While the orange genotype has lots of granules, and those are full of kind of the sepia Pterin orange color, the alba phenotypes actually lack a lot of these pigment granules. They seem to be literally colorless. They don't have the pigments in the same way. If we go in and actually remove these pigments chemically, we also recover the white color in the absence of pigments. So it looks like what's happening here in Colias butterflies is that orange butterflies are investing in making colors, and alba phenotypes are not making color. which might explain some of the energetic trade-off that we think we're seeing in terms of alba females having a higher fat body content and a higher fecundity. They just might have more resources in a simple Y model of reallocation to invest in those. Okay, how did this life history strategy evolve, right? We're going to identify the locus of Alba in this European Colias species, and then eventually get to the North American. But we have to start somewhere, and we're going to start in Europe. This was work from actually my first PhD student. Here we took 15 Alba and 15 orange females, and we did a genome-wide association study. And we're able to map the genetic basis of Alba to a single genomic region. And in that genomic region, all these white dots represent individual SNPs that are associated with the difference between orange and white. And those were all located right next to a gene called BARH1. And BARH1 got us quite excited. It's required in developing eyes of flies. It's relatively well understood in terms of its evo-devo role in that context. It's a homeobox gene. It's a transcription factor. It actually represses the developmental trajectories of other cells. So it kind of moves things along in terms of normal eye development. And when you knock it out, you get essentially a Drosophila eye that lacks pigment, right? Now that got us really excited, right? Because alba is the absence of pigment in our butterflies. So we thought that was really interesting. And then what we did was we used CRISPR-Cas knockouts of the BARH1 gene in the coding region to see what that would actually do to test this hypothesis that that was an important gene in our color phenotype. Here we're just injecting a Colias crocea egg with Cas9 and guide RNAs. This is the normal head eye of a Colias crocea female, and they're quite beautiful bugs. They have a nice, vibrant green eye. That black region you see there is just kind of an optical illusion of the structure of the eye, but it's just a nice emerald eye. And when we use CRISPR to knock out the BARH1 gene itself, it causes a dysfunction of that gene everywhere it's being used in the body. And what we see is this: the butterfly has these patches of no pigment in its actual eye. And so this is a mosaic, a CRISPR knockout mosaic, showing that yes, indeed, the BARH1 gene does play a role in butterfly eye development, as we would expect from work in Drosophila. And it's changing a green eye to the absence of pigment, which in this case is black. Now, when we did that in an alba female, we actually were able to recover orange pigmentation, right? So what we did was we think that BARH1 is acting as a repressor of scales pigment granule formation. And here we're actually able to recover that when we knock out BARH1. So we're removing its repression of pigment granule development. And here we're kind of just going in closer to kind of drive home that point. And again, this suggests that BARH1 has a novel role in pigment granule development of butterfly wings. And importantly, this is a novel gene. This is completely independent of the genes that were part of the developmental wing pattern toolkit that I talked about in heliconius butterflies. So this really is a very unique gene for pyrid butterflies and color variation, which is perhaps not surprising because it looks like it involved in pigment granule formation, which is a Pteridine color, which is not seen in the other butterflies. Right? So it's unique, and we're quite excited about that. But that was only what we knew, or we discovered that in Colias crocea, right? That was our European Colias representative. And so because we're trying to figure out if this is something shared by other species and perhaps across genera within Pieridae as a whole, we want to start moving out from this one species observation, start trying to put things into a phylogenetic perspective.
So Chris, sorry to interrupt, but so do you think that the BARH1 is the reason why the alba females, or why the alba phenotype doesn't have the granules?
Yeah.
And so do they also have this eye phenotype too?
Well, that's a great question. We're going to get to that. So when we did this in this experiment, where we were actually doing CRISPR in the coding region of the BARH1 gene, all individuals, whether they were male, orange female, or alba females, all showed the eye phenotype. Only alba females showed the wing phenotype. Yeah, so we think that of course we were knocking out BAR everywhere it could function because we're knocking out the coding region of the gene, right?
I see. Okay.
But where we're going to be going now is we're going to end up cutting what we think is the cis-regulatory region of the alba allele, and we're going to still get a phenocopy on the wing, but we're not going to change the eye at all. So we're actually going to see a much more relevant phenotypic effect once we actually get to what we think is the regulatory region of the alba allele.
Cool. Thanks.
Great. So we were able to identify the locus of Alba and Colias crocea. And now we're going to try and place this finding in a larger evolutionary context, trying to ask whether or not Alba evolved once or multiple times. Again, trying to weigh in on this idea of what is an ancestral state reconstruction and how much can we trust those. So again, Colias is comprised of about 70, maybe 80 species. We think it diversified recently. I'm going to show you evidence for that. On the right side here, we have kind of a plate of an old book of Colias butterflies. So you can see there's a lot of variation in terms of yellows and oranges and whites, albas. And so what we did was we sampled individuals from the global distribution of Colias butterflies, North and South America, Europe, Africa, Asia. We collected species with and without the alba polymorphism, and we sequenced their genomes to a pretty good coverage, and we used that to build phylogenies. So we used a gene tree approach using individual exons of genes from across the genome. And that way I was getting blocks that were not likely to have recombination within them and getting multiple independent blocks like that. I used about 9,000 exons, again, one per gene, on average about 150 base pairs. I filtered a little bit for variable sites. I initially built chromosome level species trees. So for each one of these exons, I built a gene tree, and then I brought those together within a chromosome to build a chromosome species tree, if you will. On the left, you see that for the Z chromosome. On the right, you see that for the 30 autosomes within Colias. And you can see there's a lot of alternative trees at the chromosome level across these different species. Each gene tree was built with IQ tree, and I got the species tree using the latest flavor of Astral. This is just another plot of that same autosomal species tree. And again, you can see a lot of the conflict among the trees, potentially indicative of the complex evolutionary history, the recent radiation, incomplete lineage sorting, potentially introgression across or among these different species. The row of boxes that are yellow and blue right next to the phylogeny, next to the name of these species, that indicates whether the species that we sampled, we sequenced a genome of females, was alba or orange. So you can see that we have a lot of alba and orange individuals across the phylogeny. Down here at the bottom is a sister genus representative, Zerini sasonia. And over on the right is the full set of exon datasets, trees used for one species tree. And you can see that the overall species tree for the entire dataset is relatively consistent with the underlying individual chromosome level trees. And I've colored it by region that the butterflies came from, whether it's from North America in blue, Europe in green, the whole Arctic species that are found actually in North America through northern Russia into the Scandinavian countries. There's a number of species that actually span that whole range. And those are colored in orange. And they kind of fall in a region of the tree that we would actually perhaps expect them to fall. And the South American clade is really the most divergent within the entire tree compared to everybody else, which is nicely concordant with what we'd expect. Gene concordance factors throughout this species tree, if I try and look at the support that every individual node has at the gene level or the SNP level, they're actually all quite low, which I guess is to be expected. This looks like it's a pretty quick radiation with a complex evolutionary history. That's perhaps not uncommon at this scale of evolutionary history. And again, Alba is found across all these different geographic regions. So this was the species tree framework. And what I wanted to do is take this large scale data set and then try and get a relatively unbiased and accurate estimate of when the major divergences happened here. But I still wanted to keep that within a multi-species coalescence. So I didn't want to just collapse all my nodes into a large concatenation and then run that through BEAST. I wanted to try to do something through SNAP. SNAP, working with a very large data set, is actually incredibly slow, as is StarBeast. So trying to figure out what's kind of the least biased, using the most data, still trying to estimate the underlying phylogenetic tree while bringing in a time calibration, multi-species coalescent. How do you do that? It's actually quite complicated, I think, with the current set of tools and the large data sets that we have. What I ended up using was SNAP and taking the longest BUSCO exon, so the longest exon for each of the identified single copy genes within Lepidoptera. There's about 5,000 of them that are well recognized. I ended up using around 4,000 after filtering. So I took the longest set of 4,000 exons from the single copy orthologs that should be single copy in everybody here. And I pulled out SNPs from those. What we have here is the resulting tree from that analysis, which gives us a time-calibrated phylogeny that has very similar topology, thankfully, to what I was using when I was doing the whole kind of astral species tree reconstruction. All nodes have a really good posterior support, except for the two that I have identified on the tree. I started with 2 separate trees and ran each for about 4 million iterations. Initially, I was using one tree, and I had multiple runs, and I thought it was converging. The internode length branches were incredibly compressed, and I was using maybe 4 taxa fewer. And I got rid of a lot of the somewhat redundant taxa near the terminal tips. And I think that caused a lot of collapsing of internal branches and gave me a really strange result. And I was reducing the number of taxa because, again, these runs are very computationally demanding and very, very slow. So I thought reduce the taxa, and see what I can get. I think that was wrong, and it was only after trying to find a higher performance processor and using a slightly larger data set that we could see that what we were getting before was actually wrong. So I think that the bleeding edge here in phylogenomics with trying to do what ostensibly should be kind of objective, good time tree dating is actually still relatively challenging. And I really recommend people try and really push the number of iterations they do. And the 2 starting trees that we used, one was an ultrametric tree and one was a cladogram. And they, after 2 million iterations, they really were not even converging. And that's kind of weird, right? But after 4 million, everybody conversion, things look great. So we have a lot of confidence in this tree now. Yeah, so I ended up using around 1,300 SNPs from all these different exons. And this is a slight downsampling from my full data set. And yeah, so this is a kind of shout out to Michael, Matt Shiner, who has a wonderful GitHub repository where he really walks through a lot of the details of how to go about doing a very nice SNAP analysis here, which he's used in several publications, but he really makes that wonderfully and unfortunately uncommonly accessible to the community. And so I would definitely direct anybody who's interested in these type of things towards his tutorials, as well as this specific one on how to use SNAP for a Bayesian divergence time estimation. So is Alba orthologous across Colias, right? I'm trying to bring back our focus here. And I just showed you earlier that we know the genetic basis of Colias crocea and what's causing the difference for Alba here, or at least the locus that's driving this primary difference. And we're asking, is this the same locus in North American Colias? And if it is the same locus, well, they last shared an ancestor over 1 million years ago. Right, so that would be a deep maintenance of an alternative life history strategy, right? And that's why we're excited about trying to go after this. To do that, we needed to, well, build a high-quality genome for Colias eurythmy, which we did. We did a whole bunch of packed biosequencing. And with a little bit of high-C data that quickly fell into 31 chromosomes. We also used a linkage map to do that. We did some polishing, had really good BOOSCO results. Here I'm just showing the alignment of our genome to the outgroup genus, Zerini Sassonia, showing that, yeah, it looks like it should. And here's the result of doing a genome-wide association study in our North American Colias. Across the top, we've used 15 Alba and 15 orange individuals to find a single region that's associating with Alba, and then we zoom into that region and we still have that nice peak. And then we zoom into that region around the peak. And what we're showing you here now, at this top row, is data from Colias eurythmy, where we have genomic data from an Alba individual mapping across this region. And just below that, data from an orange individual. Now, on the right side, this is where the BARH1 gene region is. And we have data from both morphs mapping very well in that region. And as we get farther to the right, you'll see that we're only getting Alba content mapping in this region, right? What we call the Alba, or a region unique to Alba, we've colored in green. We know there's a lot of repeats in this region in gray here. We annotate, we find that we can annotate them as repeat regions. So we know that's repeat content. We find nice, unique content in this pink region. So this pink region we think is quite nice. When we map data from the European species, where we know a lot about Alba, from the BARH1 region, we map all the genomic data through here. It's, again, mapping great through BAR. We get a lot of repeat content here. This depth here is just multiple copies of the repeat elements in here, just mapping here. So it's stacking up on itself from different regions in the genome. But again, we get this one region in here where only Alba data is mapping, but nothing from orange. And so we call this the Alba unique content region. It's about 1 kb. And this is our putative region that we think is harboring maybe transcription factor binding sites that are controlling when and where the Alba phenotype is being turned on. In this case, we think it might be controlling when and where BARH1 is being activated, right? And I'm going to show you more evidence for that. So it looks like Alba is the same in taxa from North America and Europe. For Europe, it maps to near the BARH1 gene. In North America, it maps to the exact same place. But what about the other Alba species in our data set? And here, I'm really going to leverage the power of having genomes for all these species. And we're really going to get back to this early question of, is Alba really Alba across other species, right? And although we eventually might want to look at this question across different genera, we're staying within Colias and at least asking, between species that last shared a common ancestor about 1 million generations or years ago, is it really the same region that's controlling Alba? And that's what we're doing here, right? So what I want to go back to real quick is keep in mind this pink region right here. This is the pink region that only had Alba unique content in North American species and European species. So we think that this is important. So the hypothesis is, if that region is causing Alba, then that region should be present in every Alba species, and it should be absent in every orange species. So we're going to look at that region in the genome from all the species in our phylogeny. And that's what I have here. We have the phylogeny, and then we have whether or not there's read content mapping in that Alba unique region from species that are white, that's all shown in blue, where they actually have content, and all the species that are either yellow or orange, they're actually not having any content mapping in the middle of that region. So this is showing a direct concordance between that unique region. Only Alba individuals have something mapping in there, i.e. they have content for that, and orange females lack it. Now, in order to put a p-value on that, we said, okay, that window is about 600 base pairs long. Let's break the entire genome of Colias eurythemi up into 600 base pair windows and map all these species genomes to that entire Colias eurythemi genome. And for every 600 base pair window, for every individual, we can ask whether or not they have content. So if you'll notice on the right side, I have numbers popping up. So for example, in this 600 base pair window that represents what we think is the Alba unique region, we can score this Colias lesbia has nothing mapping there. For that window, it gets a 0. Euxanthe, a 0. Interior, 0. Bari, 0. Pteridine, 1. Right? So we can go through and actually score this for this window in a binary way. And for this Alba unique region, there's a direct concordance between having white and being 1 or being colored and being 0. But we can score that for the rest of the genome. And when we do that, and we simply just take the difference between Alba versus orange, it's a random distribution around 0 for the nearly 500,000 windows across the genome. But only when we look at it where we're seeing Alba versus, or I guess orange minus Alba, we get a direct concordance between only Alba individuals have content there and orange do not. So genome-wide, for all these species, it's a unique content directly associated with the Alba phenotype. And so that's how we extend this Alba unique region from our 2 species to the whole phylogeny that we have here. Does that make sense? Okay, well, now we're going to test that hypothesis. So recall that we used our genome-wide association study and data from 2 species to get to this pink Alba unique insertion region, and then hypothesize that that should be the region directly associated with Alba across all species. And then I just showed you that indeed only Alba females have content in that region. Orange females do not. And that's pretty unique across the entire genome for all these species. And so this is a comparative analysis of an insertion, and we're using that to identify a putative cis regulatory element that's actually controlling Alba, right? I mean, not only can we say that this is unique Alba content, but if it is, then shouldn't that be the region that's actually turning on the bar gene to make Alba Alba? Well, we decided to test that with CRISPR. So Colias actually went in and designed guide RNAs for this Alba locus back in our model, Colias crocea, and we cut in there. Now, this is the model of bar H1, and these arrows on the top, these 2 red ones, and the second exon of bar H1, that's where we cut in the earlier study. And when we cut in the exon, every place this gene's going to be used, it's going to be defective, which is why we're getting I mutants, but we also got wing mutants. When we're cutting in the regulatory region of bar H1, we would expect to still get phenotypes in the wing, but the I phenotype should disappear. And that's indeed what we found. So we think this is good evidence that we can use this comparative phylogenetic approach of looking at what we think is the insertion region to identify, essentially do phylogenetic footprint mapping for a Cree, and then we actually tested that using CRISPR. We also looked at a lot of integration analyses. Again, these genomic data sets for phylogenetic analyses are incredibly rich, and I think a lot of the readers of SysBio know this. Here we're just highlighting more that we can do with these genomes. I mean, here we use it to come up with a hypothesis about what was driving the trait across the tree. Here we're using it to look at levels of integration between different clades in our phylogeny. And we see that it looks like there is some integration between whole Arctic species integrating with either relatives in Asia or relatives in North America. We can also look at integration using more of our phylogenetic level data, where we're trying to assign integration events to, instead of tip level dynamics, Here you can see that there's strong evidence of integration between the common ancestor of Hecla, Nasties, and Tyche here that was maybe doing some type of integration with Colias Facomini. Right? And so this allows us to say that maybe there was integration event deeper in evolutionary time, which I think is a very powerful way of looking at things. This suggests that, of course, there's been integration across Colias during their evolutionary history. That could have been how Alba was maybe moving across species. We don't really know. We definitely know there was integration. We can see the evidence for it here. But when we actually try and localize integration events in or around the Alba locus near the BARH1 gene, we actually can't find any specific signatures of integration taking place there. Okay, so in summary, it looks like this Alba phenotype, this alternative life history strategy, evolved only once. And it was at least 1.5 million generations ago, because these species all have at least one generation per year. And we've been able to temporally calibrate this back to at least 1 or over 1 million years ago. So it's many generations ago, probably much farther back in time, because Colia species in South America also have Alba segregating. And the last time South American Colias shared an ancestor with the rest of Colias around the world was probably over 3 million generations ago. So all of a sudden now, Alba, this alternative life history strategy, looks like it's quite old. It's probably moved across species by integration, hybridization. And it looks like it might also have been maintained in species by various levels of balancing selection. I didn't show you those analyses, but Colias has recently developed a nice test for balancing selection using indel frequencies across the genome to get a genome-wide estimate for what those should look like, and then compared what's happening at the Alba locus to that genome-wide estimate. And the Alba locus is much more intermediate frequency compared to the rest of the genome. That's a challenging test to do, but it shows us some very nice results. So balancing selection looks like it's important. Integration looks like it's important, but it's been maintained for a long time. It has one origin. So now we're wondering, well, how long can it be maintained as a polymorphism in a given lineage? What determines the outcome of whether it stays polymorphic or whether it goes fixed? That gets back to my initial goals of trying to bring in the eco-physiological aspects of how this Mendelian trait is interacting with the biotic and abiotic conditions that each species finds itself in. We're certainly curious about what the pleiotropic effects are of Alba, how it might give rise to these differences in the fat body and fecundity, if it really is just a difference in allocation, or whether something else is actually taking place due to BAR, or maybe there's some regulatory element within the Cree locus or near it that's affecting something far away. We don't know that yet. But to get back to the earlier part of the question and put this into perhaps a larger phylogenetic framework, does Alba exist in other genera within Pieridae? And to come back to this figure then, which is really close to where we started out, there's different Alba phenotypes in these species. And these are from different genera. And they last shared a common ancestor with Colias quite some time ago, talking 20, 30 million years. Is Alba the same in those species? To ask that a different way, is ancestral state reconstruction valid when we go really far back in evolutionary time? I'm at least showing here at the level of Colias, i.e. a genus, we definitely can go back to probably the basis of that genus and be pretty secure. But testing these things is a challenge, and it's one that we look forward to getting further into. And I hope this has made you think differently, or at least more critically about what ancestral state reconstructions may or may not mean in your system or others. I did not do this work alone, noted Kale. We're part of a larger team that we call Team Alba. That's a diverse group of great colleagues. This includes former PhD students Kale Tunstrom and Alyssa Warwick, Joseph Hanley, Pasi Rastas, Anton Shurkovin, may he rest in peace, Andy Warren, Akito Kuohara, Sean Chauvel, Vincent Firokota, Ward Watt, Adam Porter, and of course, Arnaud Martin. Thank you for your time.
Thank you, Chris. Yeah, so I do encourage people to ask questions via YouTube chat. We do have somebody who's already asked a question, Sam. He says, hi, Chris. Great talk and a very cool system. 2 questions. How did you time calibrate your Colias tree? And then also, how is this Alba trait female limited? I don't think I understand the mechanism.
Yeah. I'll take the last one first. We don't know exactly how it's being regulated in a sex-specific way. One easy way to do this would be to have a female-specific double-sex transcription factor binding site somewhere located in the Cree that we think we've identified, that in the developing wing tissue, when these pigment granules are being formed, that transcription factor would, of course, need to be there, and that could give rise to this, right? That's, double sexes play these roles in other butterflies, and it could be doing that here. We just have to generate the data to look for whether or not that transcription factor binding site is there and playing that kind of a role. We think we see hints of that, but we're not really ready to stand upon that yet. In terms of time calibrating my phylogenetic tree, the way that we did that, I'm just going to go back to there to facilitate my example, is we used an external calibration point from another study. So the outgroup was Pterin sassonia, and in, I forget exactly the study, but using around the 8 standard phylogenetic genes used in Lepidoptera, a larger sampling across Pieridae did a pretty good job of coming up with a date for the divergence between Zerini and Colias. And it was that I used as the divergence time between those as a prior when I put that into the SNAP analysis. And I used that prior as well as a normal distribution around it with probably about 1 million years of years.
Cool. I'm a little out of my league here, so I'm going to ask lots of stupid questions. I'm just interested in this connection between the fat and fecundity and the Alba genotype. Were you suggesting that it's sort of like the organism can save energy by not making the pigment granules, and so if you have lower fat, then it's sort of you're less able to it's beneficial for you to not make the granules?
Well, I mean, yeah, I guess I think the way that I would phrase it is that it's a simple, the way we're envisioning it here is that it's a simple Y model of reallocation. So during development at a larval stage, caterpillars are going around gathering all the resources they can, they're getting big and fat, and then they go into pupation. And during the pupal stage, they're going to go through metamorphosis and develop. And there, we think that Barr is playing a role in preventing granule formation. And in doing so, pigment granules don't develop, and Pteridine biosynthesis doesn't happen within the pigment granules. And because that's not taking place, the energy needed to do that is not used, and therefore that's left over. And we think that's then just itself allowing for the reproductive physiology to run at a different rate and course. Now it could be, so that's a vision of Barr H1 playing a role with the simple Y model of reallocation. Barr is not doing anything outside of the imaginal wing disc and the developing pupae. Now, Barr could also be playing a role in the abdomen and doing something actively with fat body breakdown or reproductive physiology and changing it. So it could be doing both, or it could be doing one. And up until this point, we haven't been able to test that because manipulating the gene itself means that it's lethal unless we have a very low penetrance mutation that then kind of shows a small fraction of cell lineages in the wing. So we couldn't actually get it into the germline to see if, and manipulate it, right? But now, because we can actually manipulate the cis regulatory element, and we don't see these negative pleiotropic consequences, then I think we can actually get this into the germline, and then we can start to look at what it might be doing in the abdomen as opposed to what it might be doing in the.
Okay, and I want to get to Mariana's question, but I mean, when you, so just to be clear, like when you say you saw no introgression, you saw basically no homology between the different regions that were like, I guess, implementing this strategy in the different species that have the Alba phenotype.
Okay, so you're asking about our introgression analysis, yeah?
Yeah.
Excellent. So yeah, so Collie got really into doing all kinds of different introgression analyses, and he started off doing kind of a trio analysis where he compared every species in trios within Eurasia and Africa to generate D statistics to try and look at the levels of introgression detected here. And this is the minimum significant D values that he found. And there's some introgression among these species. And again, there's some among North American species. But whenever we started to integrate or use trios that included individuals from whole Arctic taxa, the means and the range and everything went up, right? So we think there's a lot more introgression happening with the whole Arctic taxa, which kind of makes sense. A lot of the whole Arctic taxa are actually fixed Alba species, so it's a great source to kind of pump Alba into North American taxa that might either lose or not have had Alba in them. Now, here, it's a similar analysis. It's a little more elegant and allows you to kind of polarize introgression events along the phylogeny. The third way that we did it was try and actually do this type of Alba-Baba D-statistic test, but in a sliding window across the genome for all different sets of species to see if we could actually see signatures of introgression in windows as we move across the genome. And we actually didn't detect those anywhere near the Alba locus itself. So why might that be happening? We think that that's happening because Alba itself is actually a very complex region. It's composed of a lot of repeat elements, and fundamentally it segregates as an insertion deletion. So when you're doing this type of Alba-Baba test across the genome, you're using a variant calling file, a VCF file. That's essentially your SNP files. And you need to kind of filter that in a way that you're working with regions that are represented across all the individuals you're doing an analysis upon. And for Alba, because some individuals won't even have data mapping there, inherently we can't use the core region for Alba when we're integrating that with a bunch of orange species, right? Because it's just missing. So we have to filter it out. And unfortunately, the regions even flanking Alba, upwards of 10,000 base pairs on either side, those are low complexity, high repeat density regions where, again, we can't call reliable SNPs because the regions are too complex. So we have to, by default, actually get relatively far away from the Alba region before we can start getting reliable information. And in those shoulders outside of the Alba region, it looks like we don't see these signatures of introgression. We don't see the high to genus D values for balancing selection. It's just, we think either the introgression event happened so long ago that recombinations have broken things up and we just can't see a signature of that anymore. That's what we think is happening, but it's a difficult region to analyze using standard tools just because it segregates as an indel and it's flanked by repeat.
Cool. Thank you. Mariana has a question. She says, I'm curious if you can apply similar approaches to investigate host repertoire evolution or if the genetic basis of host use is too different from Alba genetic basis.
Collie's really been getting into this, and I don't want to steal his thunder, but I think that we definitely can use a very similar approach to studying a range of phenotypes. And we're actually in the process of doing that, and hopefully something will come out. But I think to kind of say a response to your question in a general sense, I'm actually going to go back to my very early slide, right? This gets back to this. That once you have genomes and you're doing phylogenetics, you don't just have to do phylogenomics. Any hypothesis that you come up with that might be involved in the trait that you're studying, that might be impacted as a consequence of the trait that you're studying, anything that might be correlated. You can analyze so much in the genome after you build a phylogeny. And all of these things are options, and I haven't even listed all of them. So can you look at gene birth-death dynamics for specific traits across a tree, whether they're associated? Absolutely. It's a very powerful thing to do. It's challenging because most genomes aren't high enough quality to infer that in a robust fashion. I think that up until we started having really long read genomes, a lot of genome assemblers probably collapse highly repetitive regions where there might be the more recent gene birth death dynamics taking place that could show these associations. So I think it's only in the years going ahead now that we're going to start seeing the potential for this to really realize cross taxa and different phenotypes.
Do we have the computational tool? I mean, so let's just imagine you had like, I mean, how many packed bag genomes do you want? Right? Like, okay, let's say you have like 100 or 1,000 or whatever. Like, okay, so now you have all those, and you want to do this like gene by gene analysis, or is that the first thing that you would reach for if you had all of that data?
It's tough. So a lot of it depends on the. I would love to have that data, and that data is around the corner, right? The Darwin tree of life for eukaryotes is turning out a lot of boring genomes because the UK and a lot of special stuff going on there, but it's great data. And so what can we do with it? Well, if you're staying within a genus, things are relatively easy because you can kind of cheat a little bit, map everything to one genome and infer things and look at read depth, and you kind of get away with it. But as soon as you start going farther and across genera, you need independent genomes. You need to kind of align them. You need to infer orthology, look at synteny, and kind of look. But that's hard. The vast majority of tools that are out there right now, you call the full protein set for each genome. You got the proteins from all of them, and you kind of do an all versus all blast, and you kind of sort it out, and then you say who's expanding and contracting across nodes in the tree. Now, that works maybe for most things, but it doesn't take advantage of the rich synteny information that's inherently in those data sets. And it relies upon the individual annotations of each one of those genomes being even moderately accurate, and our annotation tools are not that. So I think going forward, we have a lot of progress to do. I think this will happen. We're going to try and develop some of it, but it's right now it's early days.
Right. Yeah. I mean, I feel like the dream of like doing read mapping and phylogeny and all of this sort of modeling at the same time is, yeah, that's an awesome dream. Sounds hard. Anyway, I've got to run. We're out of questions. That was a great talk. Thank you very much, Chris. Yeah, it was really enjoyable to hear about a system that I don't think about at all.
Wonderful. I'm glad I could share it with you and the audience at Philo Seminar.
Great. Yeah, and next is Wade Dismukes coming up not too long. All right. See you, everyone.